Welcome to our openCARP video tutorial in which we will have a quick overview about cardiac electrophysiology. The heart is a muscle that beats around 60 and 100 times per minute. It's made of different muscle bundles that contains millions of cells. These cells are called cardiac myocytes, and they have the ability of triggering an action potential and contract. In this scheme, we have a closer look at the cardiac myocyte. We can see that it has a cell membrane that separates the intracellular from the extracellular medium. On the cell membrane, we can find different ion channels and pumps. Most of them are ion-specific, and in the cardiac myocyte we can find three big families of ion channels: calcium ion channels, potassium ion channels, and sodium ion channels. Inside the cell, we can also find a structure called the Sarcoplasmic Reticulum, which is a reservoir of calcium. Having a closer look at the cell membrane, we can see that there is a difference of concentration between the intracellular and the extracellular medium. Because of this difference, there will be a flux of ions crossing the membrane through the ion channels. There are two forces that will start this movement: the diffusion and the electrical force. Diffusion is purely based on concentration, and the ions will flow through open channels. The electrical force opposes the diffusion, and for that reason pumps will need energy to move ions from one side to the other and maintain the equilibrium. If we look at a single ion channel, the equilibrium potential is equal to the nearest potential. Now we can ask ourselves, how can we measure the flux of ions across the ion channels? To measure and quantify the flow of ions through an ion channel, the patch clamp technique is performed using a voltage clamp protocol. In this case, the voltage across the cell membrane is controlled by the experimenter, and the resulting currents are recorded. The setup consists of a thin pipe of glass with an electrode inside it filled with an electrolyte solution. This pipe will approach the membrane and clamp one piece of cell membrane, and the voltage clamp protocol will start. To isolate and measure the specific currents on ion channels, some chemical or drugs that will block some specific ion channels are used. As a consequence of opening and closing of the ion channels, we can measure a difference of potential between the intracellular and the extracellular medium, or the transmembrane potential. This is also known as an action potential. Before, we heard about different families of ion channels, and here we can see some of them. These ion channels will act at different times, giving an action potential some specific shape and characteristics, like for example its duration. Going on one scale above, we see that the tissue is made of millions of cells. The cells are mechanically and electrically connected. The mechanical junction is made of a union of proteins located at the cell membrane called desmosomes, and the electrical junction is known as gap junction. Through these gap junctions, the ions will flow to depolarize the neighboring cells. If we look at a fiber of the cardiac tissue and stimulate one side of the strand, we will have a wave of depolarization, and after a certain time, the tissue will go back to its initial state or resting potential. What is happening at a cellular level is that each cell is depolarizing the neighboring cells, triggering an action potential and propagating along the cardiac tissue. Looking at the entire heart, we can anatomically distinguish four chambers: the left and right atrium and the left and right ventricle. The initiation of the electrical activation of the heart occurs on the sinus node, which is located in the upper part of the right atrium. From there, the depolarization wave will start and continue through the right atrium and travel through the Bachmann bundles to the left atrium. The connection between the atrium and the ventricle is made through the atrioventricular node, where the depolarization wave will continue through a thin structure that is divided into the left and right bundle branches, and its lower portion will form the Purkinje fibers that will activate the left and the right ventricles. In this video tutorial, you learn about the cardiomyocyte and the ion channels. You learn about the diffusion and the electrical forces that drives the movement of ions through the cell membrane. You also have an overview of the voltage clamp experiment to measure ionic currents. You understand how single-cell ionic currents contributes to integrate an action potential and its propagation through the tissue. You also had an overview of the electric activation of the propagation.